So yeah, today I'm going to be talking about artificial intelligence and using computer vision and bee ecology conservation and also leveraging citizen science into the effort. So first, I'd like to start off with some acknowledgements. The work I'm going to talk about was funded by the USDA, NIFA program, the National Science Foundation, and then also some funding from Kansas State University, where I'm a professor. This research was a group effort. A lot of it was done and it still continues to be done by people such as my graduate student, Caleb Sowers, another graduate student, Jessica Butters, Tanya Kim, Bill Hsu, Brian McCornack are all collaborators at Kansas State, Claudio Gaetan at University of Wisconsin-Madison, and Richard Wang and Krushi Patel are at the University of Kansas. And then a lot of the data that some of this this new science is being developed from is from the Xerces Society, and I'm working closely with Rich Hatfield and Serena Jepson there. So I had the lab for pollination ecology at K-State, and work in my lab is mainly focused on applied ecology and conservation, basic and theoretical ecology, but more recently we're starting to use machine learning to develop tools to aid us in these efforts. So machine learning is a sort of a larger term that encompasses artificial intelligence and computer vision. So some of the questions that we try to tackle in our lab are mainly revolve around pollinator responses to disturbances and habitat loss. Pollinators face all kinds of threats, but a lot of the ways we manage pollinators are using sort of mimicking some of the ways natural ecosystems sort of persisted. So, for example, in the tallgrass prairie, it's maintained as prairie through burning and through grazing, by bison but also by cattle. It's also a highly threatened ecosystem. So there's, as you can see in the map here, there's only around 10% of the tallgrass prairie remaining. Luckily, here in Kansas, we have a big chunk of that. So it's very fortunate for us to be able to study such a nice remaining pristine chunk of that habitat. So the outline for my talk today, first I'm going to talk about pollinator decline and some of its drivers. And then I'll talk a little bit about crowdsourcing conservation through the use of community science and open data. And then I'll talk about research we're developing on AI and computer vision tools for pollinator ecology. And then I'll take a look ahead at what we're up against in terms of AI-based methods in ecology and conservation. So we have really good evidence for declines in species that we monitor closely, such as honeybees and monarch butterflies. But of the closely watched native bee species that we watch, we have the best data for bumblebees. So, for example, in this study by Cameron and colleagues, they looked at 8 species of North American bumblebees that were once widespread. And they found that 4 of these have been reduced to only fractions of their former range. And this includes the rusty patch bumblebee, which is now listed as endangered. And so we'll take you can't see much in these maps, but we'll take a closer look at a couple of them. So the rusty patch bumblebee, again, has been listed as endangered. And in this map, you can see that the historic range, or the current range in light gray, is greatly reduced from its historic range. And a lot of the places where we still see these species, they are, we don't see them in the numbers that we used to. So its overall range is declining, and its local abundances are also in decline. Out west, we have the western bumblebee, Bombus occidentalis, and looking at this species, which has also undergone severe decline, we can see the areas in pink in this graph show its current occupancy or current probability of occupancy and how it's declined drastically from just 20 years ago. So what's to blame for these pollinator declines? It turns out there's a lot working against pollinators right now. Some have called this a death by 1,000 cuts. So basically there are multiple interacting stressors driving pollinator decline. We have habitat loss, pathogens and disease, pesticides. Habitat loss is sort of an umbrella term that includes a lot of things like climate change, other ways that food resources are reduced for bees. So habitat loss, reduces the amount and access to food and nesting resources. Poor nutrition as a result of habitat loss makes pollinators more susceptible to disease and pesticides. Pesticides are more likely to be present where resources are limiting. And so, we have all of these things working against pollinators right now. So this map is kind of a nice illustration of what some of these consequences are for agriculture. So here, here we can see in the legend here, we can see on the x-axis we have bee abundance in cropland. On the y-axis we have the dependency of crops on bees. And so the trouble spot here is this pink area in the upper left quadrant of the graph. This indicates areas where bee abundance is low, but crops really rely on bees for pollination services. And then if you look out here in the map, we see a lot of pink on the map, and that's not good. So areas here in pink indicate that this large scale pattern where bee abundances in cropland are lowest in the areas where they're most dependent on pollination. Two things that don't go good together. So of these three factors that affect pollinator decline, three general factors, I focus mainly on habitat loss. So agricultural expansion is one sort of driver of habitat loss. Agricultural expansion is replacing a lot of the natural areas in the U.S. So in this map by Lark and colleagues, you can see the agricultural expansion is really outpacing agricultural abandonment. So as indicated by more of these warm red colors, and especially there's a lot of it going on around Kansas and Missouri as well. And one of the major sources of new agricultural lands is native grasslands. So what this basically says is that new croplands are sort of swallowing up native grasslands more than any other type of habitat out there on the landscape. And of course, grasslands are important habitat for pollinators, but then again, agriculture is important for people. We need food to eat. So there has to be a way to be able to devise new strategies for conserving bees and other pollinators while also maintaining our important food supply. So habitat loss in terms of pollinators can be defined as processes that reduce the amount and variety of food and nesting resources. And this can happen at many different spatial scales. So at local scales, we have, for example, a patch of grassland habitat, which can undergo different management strategies like annual harvesting for hay or biomass for bioenergy programs. At a landscape scale, we can also have different forms of habitat loss. So we have this is important where you have multiple patches across the landscape, and bees forage across wide areas. So these landscape scale factors, landscape scale habitat loss is also just as important as local reductions in food resources for bees. So at a local scale, we have different things that can affect the quality and amount of food nesting resources. Like I just mentioned, harvesting is something that harvesting grass for these, for hay, for bioenergy production. This is a sort of disturbance that can mimic some of the effects of fire and grazing on grassland. So this can actually be beneficial for pollinators. So harvesting can enhance floral resources for bees. It can enhance access to nesting habitat in the ground by removing dead grass material and basically opening up the ground for easier access for nesting. But then taking a little closer look at some of the natural history of bees, we find that 80% of bees nest below ground, and about the remaining 20% nest above ground. So this suggests that they may have different responses to harvesting or fire. So so in some of the work that we've done, we found that harvesting does have a beneficial effect on below ground nesting bees. So basically it does enhance food nesting resources for them. They're able to find more food to eat, and they're able to make to build the nest that they need in the ground, because the soil is opened up and they have easier access. However, the vegetation removal associated with haying can have a negative effect on above ground nesting species. And this, this removal of vegetation basically hauls away their nests at some point in the season after they've established the nest. They've hauled them away. If they have an established nest, then there's few stems for nesting remaining, and until potentially too late in the season for them to establish a nest. So this suggests that this is sort of correlated with studies that are increasingly showing that nesting habitat really must be taken into account when designing new conservation measures. And nesting habitat can be just important, if not more important in some cases, than food resources, such as flowers. So at more of a landscape or regional scale, regional land cover management is also very important. So if you think about a pollinator that's inhabiting a small patch of grassland, in a more heterogeneous landscape like the one on the left, compared to a more homogeneous landscape that's basically comprised of annual agriculture and not a lot of other natural habitat. These landscapes are going to be provide very different resources, because bees can forage over such broad areas. They're really going to be utilizing not only their local patch, but the surrounding areas too. And so we really must take into consideration the full the full amount of habitat that they're using across the landscape. So some of my dissertation work has shown that it sort of backs this up. So we have the proportion of habitat on the proportion of suitable habitat on the x-axis here, and then the number of either pollinator species or plant species here on the y-axis, and we can see that that as you have more habitat in the surrounding landscape, regardless of the quality of habitat locally, as you have more good habitat in the surrounding landscape, you see more plants and pollinator species. So habitat management is a multi-scale issue that we really have to consider all of these different spatial and temporal skills for pollinator management. All right. So so habitat loss can impact biodiversity at these different scales, but it can also affect the way plants and pollinators interact within a community. So this is sort of a diagram of a network of interactions between plants and pollinators. And we can see that, as you change the biodiversity of the system, it can affect the way plants and pollinators interact. But this, the changes in biodiversity are driven by habitat loss. So habitat loss itself can really have, multiple important effects on plants and pollinators. These interactions are really important because they can determine the stability of the system. The different ways plants and pollinators interact can either put the system more at risk or sort of make it more robust. So we wanted to look at this and see how floral availability determines pollinator foraging strategies. So the strategy of the pollinator is, really determines what plant species it interacts with. So in this example, we have a couple of different hypotheses here. So in this sort of a really simple diagram of a plant and pollinator community. We have plants that occur in either high abundances and then different species, all the way down to low abundances. And one way that pollinators may choose to forage is by preferentially targeting these highly abundant plant species and giving, very little effort to foraging on low abundant species or low rewarding species. In this case, would probably mean that competition for these resources doesn't really matter. And for conservation, this means we just need to plant a few but abundant floral species. Alternatively, plants may divide up resources in order to reduce competition. So instead of targeting abundant species, we may see plants actively foregoing foraging on some species and foraging in these smaller groups, or sometimes they're called modules. So we have these little sort of tightly grouped bunches of species with very little overlap between, and this sort of alleviates competition. Plants or pollinators can specialize on different plants. And for conservation, this would mean that we need to plant a diversity of different floral species to support all of these pollinators. And so our research has basically shown that in order to support the greatest diversity of resource or the greatest diversity of pollinators, we need to plant a diversity of plant species, because pollinators really will divide up their resources like this. So, and we find that the more plant species there are, the more different kinds of floral resources there are for them, the stronger they'll divide up those resources. So it really gives specialists, which are the most vulnerable species to habitat loss, it gives them sort of a fighting chance to be able to sort of stay in the system and maintain that highly diverse system. As the plant community sort of degrades and it gets fewer and fewer species, then that's when we start seeing the few remaining pollinators more focus on the most abundant species. So it's the pollinators sort of adjust their foraging strategy based on the local community to best provision for themselves. So what this all means really is that we need, for bee conservation and pollinator conservation in general, is we need more flowers and nesting resources out on the landscape. So what we can do is plant native flowers in field margins, use cover crops or no-till strategies. This is more in agricultural areas. In urban areas, we can plant urban gardens, roadside plantings, but we also need to think about conserving and restoring wildlands. Conversion of grasslands to annual agriculture has recently been driven in large part by demand for bioenergy and federal policy. So if we can reclaim some of these perennial grasslands on marginal croplands that don't provide for a lot of production, we can use these grasslands for a different kind of production. So for example, sustainable bioenergy production or grazing. And this should provide a lot more habitat out there for pollinators. So like I started with, we have a lot of knowledge about, you know, some species like honeybees, monarchs, and we're sort of getting there on bumblebees. But what about the rest of the 20,000 bee species in the world, and not to mention all of the other pollinators? What we really need is a lot more data to be able to map trends in both time and space. And we need new technology to help us gather data on the very large scales that are associated with these large scale, global threats to pollinators. So two things can really help with this. You know, the development of community or citizen science programs, but also AI-based data collection. And fortunately, these are both beginning to be developed and really starting to take off, both with with the public and the scientific community. So there are a number of community science portals that we can access. So, for example, this is the homepage for the Global Biodiversity Information Facility, or GBIF. It is it's a free and open access data. So it's mainly contributed. The data is mainly contributed by institutions, so like universities, museums, things like that. But some of the more public data sets are also being contributed to that. And right now, there's a lot of observations. There's well over 1 billion occurrence records, and we can see here on this map that it's represented from all over the globe. iNaturalist is another community science portal. A lot of you may have actually used iNaturalist. So users to iNaturalist can, they can log on. They can make an observation out in nature, take a photograph of a plant or an animal, log on to iNaturalist, upload the photo, create an observation, and then a community of scientists and other naturalists will work together to identify the organism. And these are important observations that scientists use to for all kinds of conservation purposes. And we're finding that iNaturalist is becoming more and more popular. So with through the years since 2010 when they first started, it's especially recently it's becoming it's sort of expanded almost exponentially here. So we have on this plot we can see that both insects in purple and invertebrates are increasing, and in summer 2020 it looks like they had over 250,000 observations per week that were being submitted. So great data sources there. iDigBio, or the Integrated Digitized Biocollections, is another citizen science portal. This is more geared toward, so what they do is they are sort of digitizing museum collections. So they go into museums, take photographs of pretty much anything in a museum, natural history museums, and make those images publicly available along with the records of the occurrences. Yeah, so it's everything from plants to fossils to insects, birds, you name it, anything in a museum, they are digitizing. Bumblebee Watch is another example it's a little bit more closely resembling iNaturalist, except this is specifically for bumblebees. So here users can, kind of like iNaturalist, users will take a photo out in nature, they'll upload the photo to Bumblebee Watch. What's different is that then they can go through a key and try to identify the species themselves, and then the sighting sort of gets taken over by expert taxonomists who are trained, professionals in identifying bumblebee species, and they will verify the image before it's put out for onto these public databases. So community data and pollinator decline, or community data is going to be really important for pollinator decline. And these assembled data sets are going to be essential for understanding trends across time and space, because no single research study could really accomplish any of this stuff alone. And for these, like I said before, these global threats are going to require global data sets. So for example, one really nice use of the GBIF data was by a PhD student named Peter Sore, where he used hundreds of thousands of GBIF observations of bumblebees to examine how climate change affects their diversity across North America and Europe. And he found that greater frequency of unusually hot days drives local extinction. And most areas of North America and Europe have seen climate-driven decline in the number of bumblebee species. So basically on this map, areas in red, more warm colors indicates areas where we've seen declines in species occurrences and the number of species in that area. And another study that's come out recently by Zatara and Azen has also using the GBIF data, but this time on all bees. They found that even though the number of observations in GBIF has increased through time, the number of bee species found each year has declined since 1990. And this is really concerning because typically with more sample effort, you find more species, not less. And so this is sort of thought to be a symptom of that global decline in pollinators. So we need to take a look at this a lot more carefully. One thing that we can gain a lot of information from this, but we also need to be really careful in how we treat these data. So we need to recognize biases in public data sets like this and statistically account for them. And both of these studies have done that to some degree. So what this map shows, and you can probably see here, is that the distribution of observations across the globe is not randomly distributed. We see a lot of observations in North America, especially in the United States and in Europe, Western Europe especially. A lot in high population centers, and some areas like in South America, we have the Amazon there, where we have some of the greatest biodiversity in the world, is not really well represented in GBIF accounts. So these are things we need to consider when we're utilizing these really important data sets. On that note, these data sets are really provide blessings, but also curses for crowdsourced data like this. So we have the sparse, opportunistically collected data, usually near population centers. This can introduce biases, and this is not just the general public contributing data. It's scientists like me. We are typically studying our subjects close to home. So I live in Manhattan, Kansas. My field sites are nearby in the Kanza. So we need to really be cognizant of this. Also, the general public varies in their ability to identify taxa. This is sort of also alleviated by using expert taxonomists to back up the identifications provided by other members of the public. But taxonomists they're a dying breed. We need to be able to find more funding for them to be able to maintain the positions that they hold. And they're not able to keep up with this increasing influx of observation. So like I said, it's a blessing that we're getting so many more observations, so much good data, but it's a curse because we're becoming less and less able to handle all of the data. So what we really need are new ways to alleviate this bottleneck, especially for specimen identification. For bee IDs, there are over 4,000 species in North America, 20,000 species worldwide. And within any given ecological study, we may find several hundred species of bees alone. Bees, some species of bees, some groups of bees, like the sweat bees, are highly diverse, yet very challenging. So they've been called taxonomically challenging, morphologically monotonous. I kind of like Wheeler's take on it, that they're the despair of taxonomists, and that's not too far from the truth. So these can be really subtle, the subtle differences between them. So for example, in this species of sweat bee, or these two species of sweat bee are they're only differentiated by the width of this line on the mesoscutum. So BID is it's time consuming, it's costly, it requires expert knowledge. It also requires lethal collection methods. So many of these bees can't be identified in the field. They need to be captured, brought back to the lab, cleaned, put under a microscope, and identified. And as a result, photo ID is very difficult because a lot of these features just can't be resolved in the photos that we take in the field. These are hard to photograph. So with my program, we are really looking into this question of can AI and computer vision help remove this bottleneck? And we think it's a very promising technology, and it's going to be transformative for entomology and bee conservation. So many of us are familiar with using computer vision for detecting objects in images or video, and this type of technology is really is become very helpful for developing things like self-driving cars. But more and more we're starting to use it in the natural sciences. So in this study by Fuentes and colleagues, they are using computer vision to detect pest species on crops, and also the diseases that result from these pests. Other studies are using computer vision in wildlife studies to differentiate individual cheetahs, for example. So cheetahs can be differentiated with computer vision by using by looking at differences in the unique patterns of their spots. All right, so how does this all work? So images are basically arrays of numbers, and so the colors in an image are seen by computers as differences in the numbers in the different pixels. So we can take this image of a bee and pass it through an artificial neural network. And this can be used to learn a lot about the image, what makes that bee, and sort of generalize across different species. So our artificial neural networks are right now the state of the art for a lot of vision applications. And so in this example here, the bee image is passed through this the first part of the network here, where the network is learning features. Then it gets those features that are learned are then passed through this this highly connected network, where in the end the weights of the connections between the nodes in the network tell us, point to what species the image belongs to. So during this sort of feature learning process, the early layers of the network are really learning, different lines and edges. Middle layers, so for example, if this was a facial recognition algorithm, middle layers in the network would learn more recognizable features like eyes and noses and mouths. And then toward the end of the network, it would recognize the highly detailed faces to be able to make the classification. So this is great for pollinator identification because it only requires a set of pre-identified images for input in model training. No additional features are required. And this is really important because the model learns all by itself. We don't have to tell it, what a wing is, what important banding patterns are shown on bees. We just need to feed it an image, tell the algorithm what is in that image, and then it learns to be able to identify that thing based on these learned features. So one of the problems with this is that neural networks are data hungry. We require thousands of images to be able to train some of these models. And that's a lot of that's driven by, especially for bumblebees, differences within species. So within a species, so for example, the yellow-fronted bumblebee can look very different depending on if it's a queen or a worker or male, but also it varies across its range. So in the Mountain West, they typically have these orange bands. As you get to the West Coast, those orange bands typically disappear. So this geographical variation introduces a lot of uncertainty into the model that has to be accounted for by throwing a lot of images at the training process. So another thing we have to think about is, so for example, the species Bombus melanopygus has two different, or actually many different morphs. It morphs from orange to black on the abdomen. Only the abdomen is pictured here. And so in part of its range, it might more represent other species than it does itself in a different part of its range. And which species it most represents will vary geographically. So in one area of its range, it's going to look like Bombus vosnesensky, whereas in a different part of its range, it might most closely resemble a different species. So we try to take this all into account by throwing 89,000 images at one of these neural network models, and in developing our project that we're calling BeeMachine. So most of the data is from Bumblebee Watch, iNaturalist, and some other sources. We use the data to train a convolutional neural network, which is a specific type of neural network. And the result is a classifier capable of identifying 36 different North American bumblebee species. And this represents most of the diversity of bumblebees in North America. What we found was very promising. The accuracy was very good. So we have for all of these species, we got about a little over 91% accuracy. If you took the top 3 predictions that the model, so the model spits out its top prediction, second most likely prediction, third most likely prediction, and then it just ranks everything from there down. If you take the top 3 predictions, the top 3 most likely things, then the correct species is going to be in the top 3 close to 98% of the time. Our model, so this is sort of a 2-dimensional view of relationships between the features that our model has learned. So what we can see here is that each point is a different image, and we can see that species are grouped because our model did a good job. So points that are closer together in this 2-dimensional plot are things that look more closely alike. And the interesting thing to see here is that not only are species grouped together, but different species that are similar looking are grouped together. So Bombus impatiens, Bimaculatus, Griseocolus, those are 3 common species that to the untrained eye can sometimes be difficult to separate. We can also take a look at where the errors are. So the error rates vary by species, and this is to some degree related to the number of images for each species that we included in the model. So some species are not photographed very much just because they are not common. Some species are not near population centers, so they're not photographed very frequently. So this could affect how accurate each species might be that you would be able to identify. But we can tell that for species with a low number of images, there's a lot of variation here. So for like Bombus affinis, the rusty-patched bumblebee had the lowest error rate, even though it had some of one of the fewest number of images. And so this is, we think, a lot due to basically the level of variation within a species. So Bombus, or the Southern Plains bumblebee, has has very little variation within the species, whereas the forest bumblebee is highly variable within the species. So for the same number of training data, we're going to see a lot more error in forest bumblebee classifications. Another source of error, just for individual images that you put in there. So it just had a harder time with crummy images, images where the bee was obscured, blurry, or both. It did worse on these. So for users of this program, uploading higher quality images where you can get the full bee in nice crisp focus, you're going to be much more likely to get a good classification from that. We can look at these data to see where we need to gain more data. So if we want to have an error rate below 10%, it looks like no species with more than 5,000 images had an error rate greater than 10%. So this is sort of the quadrant, and the species in this quadrant are what we want to target for new image acquisition to retrain and improve these models. So anybody can use this model right now. It's online. We're calling it BeeMachine. It's at beemachine.ai. So to use this, it's really simple. You just click upload your image. The most important thing to consider when using this is that, usually this is the kind of image that people will take, but what you first need to do is crop right down to the very edge of the bee. So you notice in this one how each side nearly touches the edge of the bee. You want to get right up close to it, and that will really increase the accuracy of the prediction. So once you select your cropped image, this page will pop up, and it will tell you the most likely species. In this case, Bombus flavorfrons. And then the probability that it's determined to be that species. So we can see it's very likely to be flavorfrons, much less likely to be any of these other choices. So in the pipeline here, we're working on a number of projects. So there's a lot of data available in museums and research collections, just pin specimens sitting there waiting to be photographed. So we're photographing these to develop models for all bees. And this is going to be a much better or a much easier way of acquiring images of especially rare species. So this is going to be done on images of full bees, but also just their wings. The wings contain a lot of information that could be used for classification. We're finding very promising results, even on these highly difficult sweat bees. And we're also developing a mobile app for BeeMachine, so it'll be a lot easier to take out and about and look at pictures of bees. So for BeeMachine, or for computer vision in general, one thing that's really informative and going to be really useful for developing automated sampling is image or is object detection and identification. So here we can detect an object and identify it at the same time. So what we want to do is take these algorithms, trained algorithms, put them on these tiny computer boards like a Raspberry Pi, and attach them to cameras, set them out in the field, and just let it run, and let the camera and the algorithm work together to do the sampling for us, and then we'll come back later, collect the data, and do the analyses. And so far, we're seeing very promising results, and this sort of technology we think is going to really take off and be beneficial for conservation. Toke Hoye and colleagues have have been sort of leaders on this front. They developed a study on green roofs where they set up an array of cameras. It's much easier up there because they have power sources, but they're developing these techniques to look at, to aim a camera at a patch of flowers and then be able to detect visits by different species and track those visits through time to look at differences in the day of the year that different pollinators are showing up, differences in the time of day. What we're doing here in the lab is developing sort of automated trapping methods. So this is a blue vein trap and pan traps. These are two sort of standard methods for trapping bees, but they're typically lethal. So what we want to do is instead of trapping them in there, we want to aim a camera and have this automated system going to be able to sample bees with minimal impact on them. Same thing for this. So we thought that it'd be really great to have an escape hatch for bumblebees. So instead of trapping them in here and killing them, we let them go out. We aim a camera as they come out, and we get the data we need without harming bees. So lastly here, this is also highly transferable, this technology. So Brian McCormack at K-State has programs going where he's looking at pest detection in field. So we can see this little drone. We can see the shadow of it here flying over a field, and we are able to see small insects in there. And eventually, what he's going to be doing is, because it's hard to detect pests and other insects from above, he's developing these robotic drones to sort of track along underneath the understory of some of these crops and point cameras up, be able to find pests and eventually have targeted spray regimes where you're spraying only the areas of the field that really need them, and that's going to really benefit other pollinators in the end. So with that, I'd really like to thank everybody again for your attention, and I'm happy to take questions.
Dr. Spiesman, that was fascinating. I was going to ask you about the geographical variations in colors, but you answered that. That's got to be probably one of the most difficult things to get past.
Yeah, it's very difficult, but the computer vision algorithms are very robust to that. They're able to learn very efficiently. I think part of it is throwing so many thousands of images at it. It gets a look at bees from all different angles, different lighting conditions, different contexts, and it's just able to figure it out.
All right. If you have a question for Dr. Spiesman, you'll see a Q&A chat box at the bottom of your screen. Just type your question in there. If you're watching on Facebook, you can type your questions in the comments sections, and I'm monitoring that as well. Let's begin with Margot. She has a question. When can you safely remove last year's stems, leaves, etc., so as not to disturb nest or resting places?
That's a good question. So a lot of people are recommending that things like leaves, like in your yard, to leave leaves and other things, at least to some degree in some areas, just leave them there. Bumblebees will use leaves on the ground as nesting habitat as well as other bees. So that provides really good nesting habitat. So if you have sort of like out of the way flower beds and things like that, don't resist the urge to clear them of that old dead vegetation. The longer it stays there, the better for bee nesting.
I have to say, when you early in your talk, when you mentioned that 80% of bee species nest in the ground, I was pretty young when I discovered that, out mowing one day, and ran over a bee's nest. So They didn't like that.
Yeah, yeah, they're usually they'll usually leave you alone, but yeah, mowers will tend to do that.
Anthony has a question. Have you used historic bee collections from museums, universities, and researchers in your research and data sets?
Yeah, so I've used mainly data from Bumblebee Watch and iNaturalist. There's a lot of really great photographs in there. The thing that I would like to incorporate more are these images of pin specimens, and so images from iDigBio, which are sourced from museums all over the place. Those are going to be really important for identifying the specimens that we already have in hand that we can't identify yet.
All right, Brad, it's not a question, it's a comment. He writes very impressive work, and I have to say, I agree. Sandra has a question on Facebook, and it's a very good question. I mentioned running over bees with a lawnmower. A lot of people are running from bees, and here you are. It's your professional research topic. When did you become interested in bees?
Oh, I think, during my dissertation, so I did my dissertation at Florida State back around 2010. I was, working out there. I'm interested in complexity. There's so many species of bees. They do such important work for us. They interact with their community in such, diverse ways. I showed some of those images of networks. Those are what really got me interested in bees and pollinators, is just the complexity seen in those networks, what we can learn from them, and new technology to allow us to do that is going to be really important.
I didn't realize there are 4,000 bee species in North America. Is that what you said? Yeah.
Yeah. They're highly diverse.
Yeah, that's incredible. Everett has a question about the BeeMachine app. Now, when will that be available, do you think?
So the web app is available online right now. Anybody can use it, and you actually can use that. I do it all the time. I use my phone. I just open up a browser, pull up the take a photo with just a regular camera on my phone, and then pull up the browser and upload the photo to the BeeMachine site. It's a little clunky to do on a phone because you have to go through that manual cropping thing, but you can use it right now with mobile phones. The mobile app, the dedicated app, I'm hoping to get out early this summer, at least the beta version. So something that you will not have to crop yourself, or will be very easy to do sort of on your phone.
Hey, let us know when that's available. We'll post it on our website and put it on our social media outlets.
Great, yeah.
Ellen has a question. If you are using dead specimens for species determination, how do you handle fading and shrinkage?
Yeah, that's a tough question. So bees are, unlike some insects, bees tend to retain a lot of their color for a good amount of time. So we, in our collection here at K-State, we have bees that, a lot of our bees date back to the 1950s and before. So even back to the 1800s. So those ones, depending on the bee, like you get those green metallic bees, the really bright green ones, those tend to retain their color and don't fade too much, because we keep them in these dark drawers where they're nice and protected from sunlight. Bumblebees kind of the same. Some of them are okay. Some of them are just they start getting dusty and decrepit, and we don't use those. We're focusing on the fresher looking specimen. So any new specimen that comes in, our algorithms will be able to recognize those and not use those older specimens as much, because we won't really need to look at those older specimens. We won't really need to identify those older specimens because we're just getting new specimens in.
All right, Jason has a question. Is iNaturalist data made available to you? And how about Seek, their visual identification tool? Are you able to use that data too?
Yeah, so iNaturalist data is available to anybody. Their data download portal is not super easy to find, but it's available. You can find it online, and anybody can download a large table with URLs of many of the photos contained on iNaturalist. You can sort of filter it by what group you're interested in, so that's available. I think all of the citizen science portals that I mentioned, you have that capability. Seek is part of iNaturalist, and a lot of the code that they use on Seek is available. We haven't used that code, but we have used the specimen images that they provide online, and Seek is, I use Seek. Seek is really good for identifying a lot of plant species. So when I'm out doing plant surveys, I'll use that. What it's not quite good at yet is bee species. It doesn't do as well as we need it to do for bumblebees and other bees. I think one of the reasons could be is that it's trained to identify all living things, whereas BeeMachine, for example, is much more narrowly focused on bees, so we can really not confuse the model as much by having it be an expert at everything. It's more of a specialist for bumblebees.
All right. We're a little past the top of the hour, so let's do just a couple more questions. Charmaine asks, is it better to take a video or a photo of a bee for identification? Or does it matter?
Well, it always depends on context. So a video can be good because you can extract multiple frames out of it. You know, a bee's going to be moving around, so you may be more likely to catch it at an angle that is really good for identification. You're being able to see a key feature. Photos, though, tend to be of higher resolution, so you can better resolve those features. So one thing that I do is I'll sort of sneak up on a bee with my camera in video mode, and that way I can get super close and before it flies away. So you never know when it's going to fly away. So if you're just doing stills, you might miss it. But if you have it in video and you're continuously shooting as you get closer and closer, then you can get one of those high resolution frames out of the video.
All right, let's make this the last question. Brad asks, does your software capture the location information embedded in the photo?
Currently, no, but that is something that we will do in the future. Right now, the software doesn't. So my BeeMachine software doesn't capture any sort of information from the user. Basically, you upload the photo. That's the only thing that BeeMachine takes. In the future, what we'd like to do is have the location information included with that, so that we can use that location information to better inform the model. So species are geographically located. So if we know where that photo was taken, we can rule out many species just because we know that those species don't live there.
Well, it's fascinating research, Dr. Spiesman. I'm looking forward to watching it progress. And I want to try this BeeMachine.
Great, yeah.
If I get brave enough to get close enough to bees to get a good photo, I definitely want to try that. Now that I know that there are so many species out there.
Yeah. I think I'm hoping it'll be a great tool. I'd like to get it in the hands of anybody that has a camera. Get it involved in education programs. I think it'd be a great tool for. I have a 6-year-old daughter, and she loves insects, so I'm hoping to get this sort of built into some of the curriculum for grade school kids or high school kids and beyond.
Future entomologists there.
Yeah.
Well, thank you again, Dr. Spiesman, for just a wonderful talk and for sharing your research. And thank you everyone for attending tonight's program. If you would like information about all of our upcoming programs at the Linda Hall Library, follow us on our social media platforms and visit our website at lindahall.org, where I also encourage you to sign up for our e-newsletter. Also, when visiting our website, I hope that you take the opportunity to support the library through the Linda Hall Library Foundation. Your contributions help make all of our events possible and freely available, like tonight's fascinating talk. So thank you again, Brian, and thank you, everyone, and have a great rest of your evening.
Thank you, Eric.